Students, today we are just continuing from the previous topic that we are continuing that we have studied till the previous lecture. In the previous lecture we have completed the cleavage, morulation, blastulation. In the blastulation, a blastocele cavity is being produced inside the morula, and that blastocyst is running or continuing its journey toward the bottom of the female reproductive system. The bottom of the female reproductive system, when it reaches toward the uterine cavity, then it is going to search out a proper place of attachment to the uterine wall, and the attachment of the blastocyst to the uterine wall is called as the implantation. So implantation is what? Implantation is the embedding of blastocyst into the uterine endometrium, where the process of attachment occurs in the endometrium layer only. So what is implantation? Implantation is the process of embedding of the blastocyst into the uterine endometrium. Remember, my dear child, the implantation is not only the activity which is carried out by the blastocyst only, but the endometrium is equally responsible for the attachment of the blastocyst here. So we can say the blastocyst and uterine endometrium are going to perform a joint activity for the process of implantation. So implantation is a joint activity of both blastocyst and uterus. So the blastocyst is going to attach to the uterus by its embryonal knob region. If you have remember, in the blastocyst structure, the complete layer or complete external layer is called as trophoblast, and some of the inner cell mass tissues which are having the responsibility to develop the embryo are going to attach to the region of the trophoblast in one direction only. So that region is having only the capacity to generate out the embryo only. So this is what the complete implantation process will occur in that region where the embryoblasts are present. So embryoblasts are present only in the embryonal knob region only. So the blastocyst will attach to the uterine endometrium through the region of endometrium only, through the region of embryonal knob only. Remember, the process of implantation will start after 7 to 8 days. Once again, I am just saying after 7 to 8 days means from after which time period? After the fertilization. So in human, implantation will takes place or starts about the 7 to 8 days, but the complete implantation process completed about 12 days when the blastocyst is completely buried inside the endometrial lining. So here in human, implantation takes about 7 to 8 days, means the starting of the process, the attachment of the blastocyst to the uterine endometrium will get completed there in the 7 to 8 days, but the complete consumption of the blastocyst or complete burying of the blastocyst, complete imbibition of the blastocyst in the uterine wall takes around 12 days thereon. Now moving on to the next point, the place of entry the uterine wall is being completely closed by the fibrous and the cellular plug called as closing coagulum. This is an important point. Remember, as soon as the blastocyst is get buried downside or buried inside the uterine endometrium, so that time a complete mucus plug is produced around that blastocyst, so that no other structure could enters there on, and that closing is called as closing coagulum. Closing coagulum means it's a kind of closing structure which is present around the endometrial lining where the complete burying of the blastocyst occurs. One more point you must have to take care of, that till the process of implantation, zona pellucida is present in the blastocyst. The zona pellucida is very important since it is going to prevent the implantation at the abnormal site. If zona pellucida will not be there, so the implantation may occur at any of the abnormal site, and which may lead to the abnormal pregnancies. So to avoid such kind of the situation, to avoid the abnormal implantation, what will happen? The zona pellucida is preventing or checking the implantation process at the abnormal site. And after the process of implantation, now there is no role of zona pellucida. So now zona pellucida is also being digested or also being removed thereon. Now it is having only the trophoblast and embryonal cell along with the other structures filled inside the oocyte. Okay, moving on to the next. The mechanism, how the implantation took place. To understand that implantation took place, you must have to remember in previous topic only, when we were studying about the blastocyst, that time we have find out the trophoblast or trophoectoderm periclinally divide and form the two seats of the cell. The outer seat is called as syncytiotrophoblast and the cytotrophoblast. So syncytiotrophoblast is going to release out a lytic substance, lytic enzyme. That lytic means what? To hydrolyze. So lytic enzymes are going to digest out the endometrial lining, because this is required when implantation process is there. So endometrium is completely intact, so to penetrate inside the endometrial lining, this is required to digest that endometrial lining, so that the embryo could get inside or could penetrate inside that one. So this is what. So first of all, what happened? The syncytiotrophoblast of the trophoblastic layer is going to release out some of the hydrolytic enzymes or lytic enzyme that is helping in the penetration of the endometrial lining by the blastocyst. Second thing, what happens at the point of attachment, the trophoblast shows the growth of some finger-like structures. Are you getting my point? At the point of attachment, the trophoblast is going to show the growth of some finger-like projection. That finger-like projections are called as villi, and since the villi are the part of trophoblast, that is why they are called as trophoblastic villi. So what happened at the place of attachment, the finger-like projection are grown out from the surface of trophoblast, and that is called as trophoblastic villi. The villi help in the attachment of the blastocyst as well as absorption of the nutrient. This is very much common point. Attachment के लिए बना था एक ऐसा फिंगर लाइक है, जैसे एक पैग का फॉर्मेशन होता है, पैग के थ्रू अंदर धस जाता है और वहीं से वो न्यूट्रिशन को भी अब्जॉर्ब करता रहता है और साथ में अटैचमेंट को भी मेंटेन करता है. तो यहाँ पे ये चीज़ें क्लियर हो गई आपको. सबसे पहले क्या होता है, सिंसाइटियोट्रोफोब्लास्ट कुछ एंजाइम्स रिलीज करेगा. दैट एंजाइम्स आर गोइंग टू डाइजेस्ट द लेयर ऑफ एंडोमेट्रियम और अंदर धसेगा, और अंदर धसने के लिए उसी समय क्या करता है, एक फिंगर लाइक प्रोजेक्शन का फॉर्मेशन होगा जिनको अपन क्या कहेंगे, ट्रोफोब्लास्टिक विलाई. इनका दो पर्पज है, एक तो अटैचमेंट में हेल्प करना और दूसरा क्या होता है, टू एब्जॉर्ब द न्यूट्रिएंट फ्रॉम द एंडोमेट्रियल लाइनिंग. ओके, सो इज इट क्लियर? So implantation results in the formation of uterine part of placenta. So as soon as the implantation gets completed, here placenta formation process will be started. So we cannot say the processes are waiting for one process to complete and next we'll have to start. Here one after another process is ready to be started or ready to be completed here on. So as soon as the implantation process completes, it is going to start the process of placenta formation. So implantation is result in the formation of uterine part of placenta. So because we'll see in the further lectures, the placenta is the structure which is formed by both fetal part and the uterine part. So here uterine part is being consist of the region where the implantation took place. So the part of uterus which is going to form the uterine part, the part of uterus which is going to form the placenta is called as decidua. Decidua means falls off. Why it is falls off? Means at the time of childbirth this decidua is also falling off with the delivering child. So is it clear? So implantation के बाद में क्या होगा, एक placenta के uterine part का formation होगा. That uterine part of placenta is called as decidua. ये decidua तीन layers में बटा होता है. The decidua is having the three different layer. One is called as decidua basalis, one is called as decidua capsularis, one is called as decidua parietalis. Decidua basalis, decidua capsularis and decidua parietalis. So decidua basalis is the part of decidua which present below the chorionic villi and over the myometrium. मतलब एंडोमेट्रियम के ठीक नीचे और मायोमेट्रियम के ठीक ऊपर. और कोरियोनिक विलाई, तो कोरियोनिक विलाई जैसे ही धंसता है अंदर की तरफ तो वो कहाँ तक पहुँच जाता है, टू द रीजन ऑफ मायोमेट्रियम. सो डेसिडुआ बेसिलिस इज द पार्ट विच इज प्रेजेंट जस्ट बिलो द कोरियोनिक विलाई एंड अबव द मायोमेट्रियम. Second one, decidua capsularis. Decidua capsularis is the part which is lying between the embryo and the lumen of uterus. Third one, decidua parietalis. Decidua parietalis is the part of decidua which covers the uterine surface except the place of attachment. जहाँ पे attachment हुआ है वहाँ पे नहीं मिलेगा, उसके अलावा आसपास के एरिया में पूरी तरीके से decidua parietalis का formation होगा. Now moving on to the next. Just after the process of implantation, the trophoblast is going to convert in form of the rudimentary chorion. Rudimentary chorion means, once again I am just going to say you, the specific membranes are there. Those membranes are called as fetal membranes, amnion, chorion, and chorion is the complete embryonic sac, complete membranous sac in which the development of fetus took place. So trophoblast is going to start the development of fetal membrane also. If you have remember in the previous lecture when we were discussing about the blastocyst, that time I just told you the trophoblast is not important in formation of embryo because it is not going to directly involve in the formation of embryo, but without it the embryonic development could not be completed. So trophoblast after the implantation is going to convert in form of the rudimentary chorion. Rudimentary chorion means it is also the fetal part of the placenta. So it's going to form the fetal part of placenta. As we have seen the point of attachment, the uterus is going to form the uterine part of the placenta or maternal part of the placenta. So that is called as decidua, and here the fetal part of placenta is the rudimentary chorion formed by the trophoblast. As soon as the attachment completes, the implantation complete, it releases out, it start releasing out SCG. In the previous lecture only we have seen the SCG, human chorionic gonadotrophin. A hormone is there, that is human chorionic gonadotrophin, that is used for the confirmation of the pregnancy, and the confirmation of pregnancy by the use of SCG is called that the Gravidex test. So moving on to the next, SCG is called as corpus luteum of pregnancy. In the previous lecture only we have seen the SCG is required for the maintenance of the corpus luteum also. It maintains the high level of LH also to maintain the corpus luteum. And one more important point is that the SCG is also helpful in releasing out of the higher level of the progesterone. So if progesterone is there, then it will maintain the hypertrophy of the endometrium and maintains the thickness of the endometrium. It maintains the thickness of the endometrium. Means what? Ki progesterone is required. So hence the SCG is going to maintain the level of progesterone there, and means what? It is a kind of structure which is going to maintain the continuous high level of the progesterone thereon. And remember, the progesterone is going to stimulate the release of cervical mucus from the cervical gland present in the cervix region, and that cervical gland, the mucus produced from the cervical gland are going to produce the mucus plug in the cervical canal. So is it clear? So later on, this SCG release is being taking place by the placenta only, because the placenta the formation is being completed till that time. So earlier the SCG is being released by the trophoblast, the rudimentary chorion, and when the placenta is fully formed, that time SCG will be released by the placenta itself. Okay, moving on to the next topic, our next point of the implantation. My dear students, after the process of implantation, some unusual behavior in the female is being observed, like the body temperature is found to be raised temporarily in the morning period only. Soon after in the daytime it is completely normal. The females are just complaining about the fever in the morning time. Later on the daytime the fever is completely normal, and they are just having some specific changed behavior, changed mood, like pleasure of the specific food, the pleasure and the mood swing of the female. That all are the pregnancy symptom, and if at that time we are just going to find out, or we are going to perform the pregnancy test, so that time the female will be positive with the help of SCG antiserum only. So that all events are called as morning sickness. The implantation causes the hormonal changes which temporarily rise that body temperature, change in the females of the behavior, mood swinging, the specific desire to have a kind of food. And one concept is there that you must understand: the miscarriage. Miscarriage means what? If the embryo is getting died, if the embryo is abnormal, so the endometrial lining thread offs automatically to discharge out the dead or abnormal fetus outside. And how this is occurring, or how this happens? At that time the level of SCG will get reduced down, and due to the reduced level of SCG, the progesterone level will also get reduced down, and due to the reduced level of progesterone, there will be no effect over the development of there will be no chances of development of the endometrium, and the endometrium will shred off. Okay, moving on to the next point of the topic. Moving on to the next phase of embryonic development after the implantation, as the embryo is getting attached to the site of attachment or site of implantation, the process of implantation gets over. Now the female is considered as the pregnant, and the structure which is formed with the uterus, endometrium and the blastocyst is called as conceptus, and at that time the female is having the conception. Okay, moving on to the next embryonic developmental phase, that is called as gastrulation. After blastulation, the next phase of embryonic development is called as gastrulation. Gastrulation is the process of formation of gastrula. What is gastrula? Gastrula is an embryonic stage where the presumptive germ layers are being produced. The germ layers means what? The ectoderm, endoderm and the mesoderm. So it is the formation of the, what is gastrulation? Gastrulation means the formation of gastrula from the blastula. What is gastrula? Gastrula is the embryonic stage where the germ layers are being originated. What are the germ layer? Ectoderm, endoderm and mesoderm. These germ layers have to take charge to develop the particular adult structure. Like that, they are going to make the brain, circulatory system, excretory system, reproductive system, whatever is there. So these structures has to take charge. So means if these structures are being produced, if these germ layers are being produced, so almost embryonic development is over. So important point, that is what the gastrulation is characterized by the morphogenetic movement of the embryoblast. What is the meaning of morphogenetic movement? If we are just going to focus on that term, morphogenetic, morpho means the form, genetic means formation, movement. So the embryoblast cell means what? The blastomere of the embryoblast are going to move toward organ specific area in the adult. Morphogenetic, they are just going to reach toward the future area and form the particular structure. So that is why this is called as what? Morphogenetic movement. Morphogenetic movement means what? The specific movement of the blastomere of embryoblast, so that they could take their future position in the future respective organ where they have to locate. Okay, so moving on to the next. During the morphogenetic movement, how many types of movement a blastomere can show? Just try to understand. Blastomere can show 5 types of the morphogenetic movement. The first one is called as epivolley. Epivolley means what? The movement of blastomere on the surface of embryo to form the ectoderm. What is epivolley? The movement of blastomere on the surface of embryo, to form the ectoderm means what? Epivolley, epi means upside, volley means movement, means what? The blastoderms or blastomeres will move on the upper direction so that they could produce the external surface of the embryo, that is called as ectoderm. Involley, just opposite to that, the inward movement of the blastomere so that they could produce the mesodermal and endodermal structures. So inward migration of the prospective mesodermal and endodermal blastomere. So first one, ectodermal blastomeres will move upward, that is epivolley. When the mesodermal and endodermal blastomeres move downward, that is called as involley. Involution is next type of the movement. The inward migration of the blastomere in the blastocele. The blastomeres are moving inside to form a kind of cavity here on, that is called as archentron, that we'll see later. So inward migration of the blastomere in the blastocele cavity. Invagination, in sinking of the blastula wall अंदर की तरफ धंसती, that is called as invagination. Delamination, separation of the seat of the cell from the blastula. Separation of the seat of cell from the blastula. We'll see that kind of the movement when the formation of mesoderm occurs. Here the epiblast cells are going to delaminate from the surface and going to spread in between the endoderm and the ectoderm. Okay, moving on to the next point, the archentron cavity. Archentron cavity is present inside the gastrula. The cavity present inside the gastrula is called as gastrocele or the archentron cavity. That gastrocele or archentron cavity is going to give rise the future alimentary canal, that is going to form the digestive system. So archentron is what? It is a kind of cavity present inside the gastrula which is going to give rise the future alimentary canal, and it opens outside through an opening that is called as blastopore. And my dear child, we have so many times discussed the blastopore opening. Sometime may give rise the mouth and sometime they may give rise the anus, because that is the opening of alimentary canal. So when they are just going to give rise the anus first, that time the animals are called as proterostome, and when they are just going to give rise the mouth first, when they are just give rise the anus first, that time they are deuterostome. When they are just giving rise the mouth first, that time they are called as the proterostome. So is it clear to you all? Now moving on to the next point. Now, moving on to the mechanism of human gastrulation, how the gastrulation process occurs in the human. The first of all, the ICM inner cell mass, if you remember the embryoblast cell which are present in the embryonal nerve, they are called as inner cell mass. They are going to differentiate in form of two type of the cell, epiblast the upper layer and hypoblast the lower layer. Epiblast is going to produce the ectoderm, mesoderm and endoderm, means epiblast have responsibility to produce all type of the germ layers, while the hypoblast is going to form the yolk sac and the chorion, that is what the fetal part of the placenta. Okay, moving on to the next, how the endoderm is produced. So formation of endoderm. So first of all, the differentiation of the cells of embryonal nerve will occur. Differentiation of embryonal nerve means what? They are just going to differentiate in two layers, that is means epiblast and hypoblast. The upper layer is called as epiblast, the lower layer is called as hypoblast. After differentiation of them, epiblast is going to multiply and differentiate again and again, multiply and proliferate again and again. Due to the increased number of the cell in the epiblast, they are spreading, and due to their spreading, they are forming, or they are just going to provide a line, or they are going to provide a covering below the trophoblast. What happens? Due to the differentiation and multiplication of the epiblast, the epiblast will start stretching continuously inside the trophoblastic layer and will form the endoderm. Are you getting my point? It's very easy process. The differentiation, first of all occur differentiation. Due to the differentiation, now epiblast undergo the proliferation and increased or stretch in form of the seed inside the trophoblast and form the endoderm. So now the endodermal tube is also being produced, that is called as archantron, that we have discussed earlier only. What is the process of formation of embryonic disc? Next structure is the embryonic disc. How embryonic disc is being formed? One important event is that the formation of embryonic disc. What happens? Due to the continuous absorption of the nutritive fluid from the uterine endometrium, the epiblast cells are getting stretched again and again. Due to the stretching of these cells, the cells are getting elongated. Now they are not the flattened cell, they are elongated, and due to the elongated embryonal cell, due to the stretching of the embryonal cell, the embryonal cell are getting elongated and converted in form of the columnar cell. So when they are just converted in form of the columnar cell, so that region of the columnar cells are called as embryonic disc. What is there? The continuous absorption of the nutritive fluid from the endometrium, stretching of the embryonal cells, and the cells now become elongated and the columnar, and the cells, those are columnar and elongated, are going to form combinedly the structure that is called as embryonic disc. My dear child, now you can will see with the help of diagram also, now you can understand. Now the endoderm is produced just below the trophoblast. So endoderm and trophoblast combinedly forming the primary yolk sac, once again the part of fetal membrane. Endoderm and trophoblast combines to form the primary yolk sac, and similarly just above that the amniotic cavity is produced in between the embryonic disc. Embryonic disc is now produced by the elongation of the cells of the embryonic by the elongation of the cell of the embryonal nerve. So just above the embryonic disc, an amniotic cavity is present. That amniotic cavity is present in between the trophoblast and the एम्ब्रियोनिक डिस्क. तो दो तरीके की स्ट्रक्चर का फॉर्मेशन हो गया. ट्रोफोब्लास्ट और एंडोडर्म मिलके बनाते हैं प्राइमरी योक सैक, और एमनियोटिक कैविटी का फॉर्मेशन हो जाता है ऊपर की तरफ इन बिटवीन द ट्रोफोब्लास्ट एंड द एम्ब्रियोनिक डिस्क. रिमेंबर, ये दो इंपॉर्टेंट फीटल मेम्ब्रेन का फॉर्मेशन हो चुका है. दिस टू इंपॉर्टेंट फीटल मेम्ब्रेन्स आर बीइंग प्रोड्यूस्ड टिल नाउ. मूविंग ऑन टू द नेक्स्ट प्रोसेस आफ्टर फॉर्मेशन ऑफ एम्ब्रियोनिक डिस्क. Now the next one is the mesoderm layer. How the mesoderm is going to form? The embryonic disc is now produced. The embryonic disc will take the charge of formation of ectoderm and the mesoderm only. So the hind region of the embryonic disc, or the caudal region of the embryonic disc, the posterior region of the embryonic disc, will start the process of proliferation. Due to the continuous division, the number of cell will increase there in the caudal region of the embryonic disc, and due to the increased number of the cell in the embryonic disc region, they are just going to detach out from the embryonic disc and prepare a kind of seat in between the trophoblast and the endoderm. Are you getting my point? Cell of the caudal end of the embryonic disc starts their proliferation. Due to the proliferation, the cells are going to detach out from the embryonic disc and going to spread in between the trophoblast and endoderm. That is what going to form the mesoderm. The mesoderm is produced in between the trophoblast and the endoderm. Last layer is ectoderm. So remaining part of the embryonic disc, remember, the posterior region forms the mesoderm, the anterior region will now form the ectoderm. So remaining cells of the embryonic disc spreads over the mesoderm to form the ectoderm. So all three layers are being produced. The endoderm produced first, now the mesoderm, and the third one is the ectoderm. One important point is their extra embryonic coelom. It is the space present in between the chorion and amnion. What is extra embryonic coelom? It is a space between chorion and amnion. It is early embryonic stage. It is present in the early embryonic stage. So extra embryonic coelom means what? coelomic cavity एक vacant space है जो एम्ब्रियो के बाहर की तरफ रहता है. एक्स्ट्रा एम्ब्रियोनिक सिलोम, रिमेंबर एक्स्ट्रा आउटसाइड एम्ब्रियोनिक, एम्ब्रियो, सिलोम मीन्स बॉडी कैविटी और कैविटी. सो दिस इज अ कैविटी विच इज नॉट प्रेजेंट इन द embryo, but it is present outside of the embryo. So this is the space which is present between the chorion and amnion in the early embryonic stage, and it is originated from the extra embryonic mesoderm. It is originated from the extra embryonic mesoderm. So just take a look on the diagram by which we can understand well. Here you can understand the complete embryo is there. The peripheral layer is going to form the trophoblast. This blue colored cells are going to form the inner cell mass, and from the inner cell mass the lower region is going to produce the presumptive endoderm. The presumptive endoderm, the green color region, is going to show the endoderm. Here the continuous absorption of the fluid from the uterine endometrium, the cells of embryonic embryonal knob is going to elongate and form the embryonic disc, and a cavity is being produced in between the trophoblast and the embryonic disc, that is called as the amniotic cavity. In future that will form the embryonic cavity, and here the endoderm is there that will start forming its proliferation so that it could form the proper layer under the embryo only. Here the final stage of the gastrula is there. This is the final gastrula where you can see what is there, that is embryo. This layer is embryonic disc. The cavity which is present earlier only, that is called as amniotic cavity. These cells which are forming the amniotic cavity and amnion, that is called as amniogenic cell. This complete green color of the cells are spreading like that to produce the endoderm. Here the amnion, the chorion, these two are the fletal membrane. The cavity present inside the endodermal tube is called as yolk sac. Just before some time we have discussed a cavity present in between the endodermal tube and the endoderm and the trophoblast are going to form the primary yolk sac. So here this is the yolk sac, this is ectoderm, endoderm, yolk sac, and this complete red color region or the pink colored region is going to form the mesodermal region. Extra embryonic coelom lying outside of the embryo, this is the part of embryo, ectoderm, mesoderm and endoderm. Here there is no embryo, so that portion which is vacant is going to form the extra embryonic coelom. So is it clear to you all? This is what the gastrulation and the process of gastrula. So what is gastrulation? Gastrulation means the formation of the germinal layers, ectoderm, mesoderm and the endoderm. Ectoderm means what? The layer which is present at the top, mesoderm the middle layer, endoderm is the innermost layer, and all these three layers will be formed by the epiblast layer, which is being formed by the differentiation of the embryonal knob, the ICM. The two cells are there by the differentiation. Two types of the cells are produced. One type of the cell is called as epiblast, another one is called as the hypoblast. Epiblast will take charge the formation of all the germ layers, means ectoderm, endoderm and mesoderm. And thirdly, you must take care of that hypoblast is going to form the yolk sac or the fetal part of the placenta. And here the first germ layer which is formed, that is endoderm, and afterward the embryonic disc is formed. Embryonic disc is going to produce two types of the structure, and the above of the embryo is called as amniotic cavity, and below of the embryo the trophoblast and endoderm fuse to form the yolk sac that will give the nutrition for the developing embryo. All these three structures being produced. Now the complete germ layers are produced, ectoderm, mesoderm and endoderm. Means what? Now this is ready to undergo in the next process. The process is that these structures will have to perform the process of differentiation so that they could form the future organs into the adult structure. So now just take a look on the process. The next process that we are just going to discuss is called as the organogenesis. Okay, so organogenesis, my dear child, organs, genesis, formation. So dear students, organogenesis means the process by which the organs are being produced from the germ layers. Till now we have completed the process of development of this germ layer, ectoderm, mesoderm and endoderm. Due to these germ layers, now these germ layers have to reach to their particular site and will start differentiating in form of their particular structure, whether they have to form the brain, whether they have to form the stomach, whether they have to form the heart, or they have to form the kidney. All these structures will be produced now from all these structures. That is called as what? The organogenesis. So organogenesis can be studied like the fate map of the germ layers or fate of the germ layer. Sometimes this may be asked. So if you are appearing for the NEET examination, so you must go through cramming this concept, because many of the time they are going to ask the question, this organ is originated from which germ layer, of which origin, whether it is of endodermal origin, whether it is of mesodermal origin, whether it is of ectodermal origin. So here I'm just going to list out some of the structures by which you can go through that. So first layer, that is ectoderm. So ectoderm is going to generate basically the peripheral structure. So epidermis, cutaneous tissues, means epidermis and the other tissues lying closer to the epidermis, all the glands attached to the epidermis, they all are being produced from the ectoderm only. The nervous system, important point, the spinal cord and the brain is going to produce from the ectoderm. This is important point, remember. The eyes, but not complete eyes, only retina, lens and cornea of the eye are going to be produced from the ectoderm. Nasal epithelium, internal ear and the external ear, not the middle ear. Internal ear and the external ear are produced from the ectoderm. Pituitary gland, pineal gland and the medulla part of the adrenal gland. Adrenal gland has two parts, adrenal cortex and adrenal medulla. So adrenal medulla is being produced from the ectoderm. So once again I am repeating, epidermis, nervous system means spinal cord and the brain. Eyes में retina, lens and cornea. Nasal epithelium, internal ear and the external ear. Pituitary gland, pineal gland and adrenal medulla are produced from the ectoderm. मेसोडर्मल ओरिजिन, मेसोडर्म से क्या क्या बनता है? सो मेसोडर्मल ओरिजिन, एपिडर्मिस के नीचे का जो भी पोर्शन होगा, सबक्यूटेनियस पोर्शन जो भी बनेगा वो मेसोडर्म से बनेगा. डर्मिस, ऑल द मस्कुलर टिश्यूज, कनेक्टिव टिश्यूज, एंडोस्केलेटन, vascular system. Vascular system means blood, heart, everything. Blood, heart, blood vessels, everything. Vascular system, blood, heart, blood vessels. Kidney. Gonads, the reproductive gonads means what? Testis and ovary both are mesodermal origin, you know very well. Coelom, choroid and sclera of the eye. We have seen the eyes may retina, lens and cornea produced from the ectoderm, and here choroid and sclera of the eye is going to produce from the mesoderm. Adrenal cortex, medulla of adrenal gland was produced from ectoderm, and the cortex region of the adrenal gland is produced from the mesoderm. And spleen, the important structure. Moving on to the third point, that is called as the endoderm. So what are the structures produced from the endoderm? The gut means the alimentary canal or the digestive system. Digestive canal is formed by the endodermal origin. Visceral organs, all the internal organs which are associated with that alimentary canal. Gastric gland, the glands present in the stomach. Intestinal glands present in the intestine. Tongue. Respiratory system, where lungs, trachea and bronchus. Urinary bladder, kidney mesodermal origin का था, urinary bladder endodermal origin का है. Gills in case of aquatic organism. Liver, spleen was of mesodermal origin and liver is of endodermal origin. Pancreas, thyroid gland, parathyroid gland, thymus gland and eustachian tube. Remember, the internal and external ear was produced from or originated from the ectoderm only, but here in case of the eustachian tube, that is the part of the middle ear cavity, is being produced from the endodermal region. Once again I am just going to repeat out all these things because these are very important. एक्टोडर्म सरफेस लेयर के जितने भी स्ट्रक्चर्स होंगे उनका फॉर्मेशन करेगा, जैसे एपिडर्मल टिश्यूज और क्यूटेनियस टिश्यूज जितने भी होंगे. तो एपिडर्मिस, नर्वस सिस्टम इंपॉर्टेंट क्वेश्चन होता है. नर्वस सिस्टम के में क्या क्या बनेगा? ब्रेन और स्पाइनल कार्ड. आइज के केस में रेटिना, लेंस और कॉर्निया. nasal epithelium, इंटरनल ईयर और एक्सटर्नल ईयर, पिट्यूटरी ग्लैंड, पीनियल ग्लैंड और एड्रिनल ग्लैंड का मेडुलरी रीजन। मीजोडर्म से क्या बनेगा? सबक्यूटेनियस रीजन मींस व्हाट? द डर्मिस। मस्कुलर टिश्यूज, जितनी भी मसल हैं सबके सब मीजोडर्मल ओरिजिन की हैं, जितने कनेक्टिव टिश्यूज हैं वो सारे मीजोडर्मल ओरिजिन के हैं। ऑल द एंडोस्केलेटल स्ट्रक्चर्स ऑफ द मीजोडर्मल ओरिजिन। वेस्कुलर सिस्टम का कंप्लीट फॉर्मेशन, वेस्कुलर सिस्टम या सर्कुलेटरी सिस्टम का कंप्लीट फॉर्मेशन इसी से होता है, मतलब ब्लड, हार्ट और ब्लड वेसल तीनों मीजोडर्मल ओरिजिन के हैं। किडनी का फॉर्मेशन मीजोडर्म से होता है। गोनेड्स मींस टेस्टिस एंड ओवरी मीजोडर्मल ओरिजिन से बनेंगे। सीलोम, बॉडी कैविटी, बहुत इंपॉर्टेंट कॉन्सेप्ट है। सीलोम के केस में आप जानते हैं द ट्रू सीलोम इज द सीलोम विच इज प्रोड्यूस्ड बाय द मीजोडर्म ओनली, सो मीजोडर्म से बनता है। कोराइड एंड स्क्लेरा ऑफ द आई, एड्रिनल ग्लैंड का कॉर्टेक्स रीजन, स्प्लीन। फिर एंडोडर्म से क्या क्या बनेगा? पूरा एलिमेंट्री कैनाल। विसरल ऑर्गन्स, ऑर्गन्स एसोसिएटेड आर लाइंग इनसाइड द विसरल कैविटी। गैस्ट्रिक ग्लैंड, इंटेस्टाइनल ग्लैंड, टंग। रेस्पिरेटरी सिस्टम में लंग्स, ट्रैकिया, ब्रोंकस, अपन गिल्स को भी इसमें इन्वॉल्व कर लेते हैं। यूरिनरी ब्लैडर, लीवर, पैनक्रियाज, थायराइड, पैराथायराइड, थाइमस एंड यूस्टेकियन ट्यूब। So all these structures are being produced from the endodermal tube. So this is what the feet map of the germ layer are the process of organogenesis. Remember, sometime one organ develop and that organ is going to stimulate the development of another organ. So that is called as induced organogenesis. Sometime what happen, one organ is developed and that organ is going to stimulate the development of another organ, that is called as induced organogenesis here. And sometimes they are just going to provide the positive feedback and sometimes some organs are going to provide the negative feedback. Positive feedback means what? If one organ is developing, so that organ is going to stimulate the development of another organ. Sometimes some organs are going to develop and they are going to suppress the development of another organ, that is called as the negative feedback. So this is what we have to understand, this is what we have to learn. One more point, कभी कभार क्वेश्चन आता है न्यूरुलेशन. Neurulation is the formation of neural tube. So neural tube is the structure from which the future nerve cord will originate और उसी nerve cord से brain और spinal cord का formation होगा. तो ये भी ध्यान रखेंगे, ये भी organogenesis का portion है. न्यूरुलेशन प्रोसेस पूछा जाता है, तो न्यूरुलेशन इज द प्रोसेस ऑफ फॉर्मेशन ऑफ द न्यूरल ट्यूब और इसी न्यूरल ट्यूब से फ्यूचर में नर्व कार्ड का फॉर्मेशन होगा और उसी नर्व कार्ड से ब्रेन और स्पाइनल कार्ड का फॉर्मेशन होगा. तो दैट इज आल्सो ऑफ द एक्टोडर्मल ओरिजिन. सो फॉर टुडे वी हैव कंप्लीटेड व्हाट द प्रोसेस ऑफ इम्प्लांटेशन. द एम्ब्रियो इज नाउ implanted well inside the uterine endometrium. After that we have seen the complete conversion of the blastocyst in form of the gastrula, where we have seen the formation of ectoderm, mesoderm and endoderm. Finally we reached on to the process of organogenesis, where all these ectoderm, mesoderm and endoderms will differentiated, they are just going to form the permanent structure of the future embryo or future ऑर्गेनिव्स, दैट इज कॉल्ड एज ऑर्गेनोजेनेसिस, फॉर्मेशन ऑफ ऑर्गन. यहाँ पे हमने देखा फेट मैप हर लेयर से कौन कौन से स्ट्रक्चर्स का फॉर्मेशन होता है. एक्टोडर्म क्या स्ट्रक्चर बनाएगी, एंडोडर्म क्या बनाएगा, मेसोडर्म क्या बनाएगा. सो दिस इज ऑल अबाउट फॉर टुडे. In the next lectures we'll discuss about the fetal membranes and the placenta, and then we'll move on to the childbirth, because the complete embryonic development is over. Only we have to we have to understand how the embryonic development is assisted by the external factors, like how these fetal membranes are being formed, how they are helping in the development of the embryo and maintenance of the embryo, and the placenta. So these two structures we have to understand: placenta, fetal membrane, and finally the childbirth. So till the time, thank you, thanks for the class, thank you.